With that, I appreciate the opportunity for you being here, and I'm going to introduce Gregg Sanford, who is our first speaker. Gregg is an associate scientist in the Department of Agronomy here at UW-Madison. He studies biofuel cropping systems and their sustainability. His research seeks to understand trade-offs and the ability of these systems to provide ecosystem services, which include both provisioning services such as biomass production and supporting services such as carbon sequestration and nutrient retention. So welcome, Gregg.
Thank you. I do not need to use the mic, correct?
Correct, yeah.
Great. I'm going to hide the floating controls. And will this work?
Yep, it should. We needed to click on this slide and then do it.
Okay, great. Thank you. There is no longer a Department of Agronomy. Just a quick update. It's now the Department of Plant and Agri-Ecosystem Sciences. Agronomy and Hort merged. I've been asked to talk about soil carbon, which is something I think about a lot. My hope as kind of like the first talk of the day is just to maybe give a little bit of a global perspective on soil carbon, why it's important globally and locally. And then, of course, I'm an agroecologist, so I'm going to bring it back to farming, with just some general ideas and thoughts that I hope actually dovetail with some of the follow-on talks. I know Aaron's talking about regenerative ag later, Randy's talking about grasslands, et cetera. So quickly, this is not a farm audience, but every time I talk to a farm audience, the first question I get is, what's the difference between soil organic matter and soil organic carbon? So, let's just start there. Both are from plant origin, and I've highlighted plant because, as Mary says, plants are doing all the work. Any animal contribution to soil organic matter was first plant, even if that animal is us decomposing in the soil. Soil organic matter is really the breakdown products of that plant and animal material. Carbon, hydrogen, and oxygen are the primary elements in it. Sulfur, nitrogen, and various minerals are there. Whereas when we're talking about soil organic carbon, which I'm going to use SOC for the rest of the talk, we're talking about elemental carbon. We estimate or evaluate them differently. For soil organic matter, we use something called loss in ignition, which is pretty straightforward. You take a little bit of soil, you stick it in an oven at 360 degrees, and everything that doesn't burn off is ash or mineral content. And so we can take the mass of the soil minus that mineral content and ascribe the rest of the mass to organic matter. It's a little bit more complicated than that, but that's essentially what we do. For carbon, we use dry combustion, which is a much more sophisticated approach. It's hotter, but we actually directly measure carbon dioxide, which we can then convert to carbon. And in terms of their relationship, there was a recent, well, it's not so recent actually, I'm getting older. In 2010, Pribble looked at kind of conversion factors for organic matter to carbon, because forever, everybody has been using a 1.72 conversion factor. And Pribble points out that in most cases, in most places around the globe, in most soils, 2 is the best conversion factor. So it's pretty easy to get between the two. If you want to get from soil organic matter to soil carbon, you can, estimate just dividing soil organic matter by 2. And if you want to get from soil organic carbon just back to soil organic matter, you can multiply by 2. There's going to be some slop, but there you go. And then when you think about kind of the concentration in ag soils, again, these are very general numbers, but most ag soils range in the kind of 3 to 6% organic matter range. If you send a soil sample into the UW lab or Rock River lab, or one of these labs are going to do this loss on ignition, probably most of your samples are going to come in this part of the world in that range, which means that our ag soils tend to be somewhere between 1.5% and 3% soil organic carbon presently, and I'm going to get to that. Soil organic carbon is dynamic. There's a lot of batting around the term sequestration, which gives me the heebie-jeebies, because sequestration reminds me of a monk cloistered in a cell for all eternity, and that doesn't happen with carbon. All soil organic carbon starts with photosynthesis and carbon dioxide in the atmosphere. So it's autotrophs, the kind of engines of our bioeconomy that do the work. So just, if you'll bear with me with this little cartoon, we have our corn plant, and we have this hypothetical, theoretical soil organic carbon pool. Carbon dioxide is absorbed through photosynthesis by the corn plant. The biomass produced is the net primary production, the total biomass produced. As that corn is harvested or begins to decompose or residues are returned, we get plant inputs that becomes kind of unprotected carbon or unprotected detritus in the soil. This could also go for, like manure applications or biochar, which I know will be talked about today. And then that unprotected detritus kind of goes through various transformations in the soil. It can go directly into semi-protected pools, whether that's some sort of biochemical protection. For example, lignin's a pretty complex molecule, and it can persist a little bit longer because not all microbes can get at it. Or it can be physically protected either through occlusion, which is when clays and silts bind around a piece of carbon and protect it from microbial decomposition, or small particles or dissolved carbon can be adsorbed to clay mineral surfaces. But I want to just point out that this is a far less common pathway in the soil than a pathway that begins with microbial mediation. So the vast majority of carbon that persists in the soil are microbial residues or microbial mediated products. We used to have this idea that carbon entered the soil, and over time, it went through all these reactions and ended up in these things called humic acids, humic acid, fulvic acid, et cetera. Those are really artifacts of the lab methods that we use to assay them. And now that we have much fancier things and we can actually look at what's happening below ground with NMR, et cetera, we realize that the oldest carbon tends to be simple carbon, as the youngest carbon tends to be simple carbon, and they tend to be of microbial origin. So carbon comes in, microbes work on it, it's an energy source for them. The microbes have different efficiencies in how they eat that carbon. Some of them are less efficient, and so more carbon dioxide is released back to the atmosphere and less is put into their microbial biomass. Some are more efficient and more carbon is put into that microbial biomass, but it's kind of the microbial machine that then ends up building over time the soil organic carbon store. And here, I'm thinking of, this picture is primarily bacteria looking things, but you can imagine mycorrhizal fungal associations as well, taking photosynthetic carbon and exponentially delivering it to parts of the soil that the plant roots are not getting to. So, okay, then the microbes, as they chomp away at the carbon, are respiring CO2. So this is heterotrophic respiration. And it's a complicated process. It's mediated by a lot of different things. It's mediated by precipitation and temperature. It's mediated by soil texture, soil mineralogy, particularly if sorption to clay mineral surfaces is one of our stabilizing mechanisms. It's mediated by the nutrients available in the soils, and then again, the microbial carbon use efficiency, which is becoming more and more apparent as a driver of carbon accumulation or loss. And then, of course, it's mediated by what we do to the soil, how we manage the soil, what kind of crops we're growing, or litter is being returned, both quantity and quality. But at the end of the day, the amount of carbon in the soil is a pretty simple formula. Any change in carbon or stock of carbon is the difference between net primary production and heterotrophic respiration. Okay, did you have a question? It looks like no? Okay. Soil organic carbon provides a whole suite of ecosystem services, both at a local and global scale. At a local level, it's often considered the most important soil health indicator, or in some recent scientific manuscripts, kind of the foundation or bedrock of civilization, if you will. It's a little hyperbolic, but certainly if you look at any soil health study that tries to correlate different soil health parameters, you'll find that the vast majority of them correlate with carbon, and that carbon is one of the best indicators of soil function. The Soil Health Institute actually did this project called the North American Project. That's an awful acronym, NAPSHM, North American Project to Evaluate Soil Health Measurements. And they looked at 124 different long-term sites from Canada down to Mexico. And their kind of takeaway was like, they threw everything in the kitchen sink at these sites to see which soil health indicators were the most responsive. And their take home was that total carbon was the principal one. And then if you want, you might add something like particulate organic matter and microbial respiration. But carbon was kind of like the crux. What does carbon do for our soils? Well, if I'm a farmer, I'm excited about building carbon because it's going to improve my soil structure, which in turn improves the aeration or gas flow in the soil. Building structure helps provide erosion control because you have aggregates that are sticking together, provides water regulation. This is really interesting. As structure improves, organic matter increases, you increase the water holding capacity of the soil, so the ability of your soils to hold on to the rain that just came, but you also improve the drainage of the soil, the ability of those soils to drain should they be inundated or waterlogged. So with structure comes a lot of great water regulation. Cation exchange capacity. Cation exchange capacity is the capacity of a soil to exchange base cations, which are plant nutrients, calcium, magnesium, et cetera. A lot of it, and most of us in soils learn about the importance of clay minerals, negatively charged clay minerals, as this huge bank of cation exchange sites. Carbon has just as many, if not more, and so building carbon increases the cation exchange capacity of your soil. And then as microbes chew on the carbon that's building, nutrients are released. Remember, organic matter has a lot of sulfur, nitrogen, et cetera in it. And so the more organic matter you have, the more nutrient cycling and potentially available nutrients for the plants. Okay, what about a global level? Sorry, I had all these really cute icons. It doesn't matter. They're hidden behind those things.
I can try to
just pretend that they're there, and they're really cute. Globally, and these are super rough numbers, and they depend on who you are, like what assessment of global carbon stocks you're reading. But globally, there's about 47,800 petagrams of carbon, give or take a petagram. Petagrams are big. 1 petagram is 1 billion tons, or 1 metric tons, or 1.1 billion short tons, or 192 Great Pyramids at Giza. So that's 1 petagram. They're big. So of the global carbon pool, 2% resides in our atmosphere as carbon dioxide, methane, chlorofluorocarbons, whatever the carbon is, but about 2% are 760 petagrams. The biotic carbon, which is us, trees, squirrels, whatever, is about 560 petagrams, or 1%. And there's, of course, an exchange here. Photosynthesis brings carbon into the biotic pool, plant respiration sends it back out to the atmosphere, microbial respiration, those types of things. The soils pool is about 8%. So this is where I'm hedging a little bit. The estimates range from 5 to 8. I'm going with 8. Soil carbon, about 3,950 petagrams, 75% of which is organic carbon. So the other 25% is inorganic carbon. Think chalk soils or calcium carbonate, dolomitic limestone that's out in our driftless area. So the vast majority of our carbon pool is organic carbon.
I was just curious, why do they measure it in grams if it's such a huge quantity?
That's a great question. We could talk about metric tons, but I think putting it in petagrams enables us to get the number down into the thousands as opposed to some sort of exponential number. Yeah. Okay, so then we have soil carbon, 75% of which is organic. Clearly, soil carbon is interacting with the atmosphere based on that kind of how we build carbon diagram we looked at through soil respiration. Now, The reason that soil organic carbon is so important, at least in my mind, but also in the mind of all the people trying to capitalize on carbon, is because relative to the pools that we can manage, it's pretty big. So the atmosphere is 2% of total carbon on the planet. The soil is 8%. All of the trees and plants and animals are 1%. You know, our land management can send carbon from the biotic pool back into the atmosphere, right? Like burning. There's lots of ways we could do it, but so too can our soil management, right? We can till soils, oxidize carbon, send it back up into the atmosphere as carbon dioxide. And the fact that this pool is so big relative to this pool means that we've got a bigger lever on the atmospheric CO2 concentration. That's not all the pools of carbon. We've only dealt with like 10% of the total carbon on the planet. The kind of huge pool is in our ocean. 80% of the carbon in the world is in the ocean. But the real gorilla in the room, I think, is our fossil carbon pool, which is set aside. It's a geologic carbon pool. It originally was a biotic pool that then became a soil pool and now is kind of locked away. It's as big as the soil pool, and we seem to be really happy burning it. So the fact that we have 9% of the world's carbon potentially to emit to the atmosphere is another huge lever, and I think part of the reason why we're having these conversations about circular bioeconomies. Soil organic carbon is distributed, very randomly to some degree, but not randomly globally. We find a lot of carbon, actually the most carbon, in our boreal forests that are moist. This is a key piece here. Second kind of biggest pool, 210 petagrams, is in our cool, temperate regions of the world that are moist. Again, the water content or moisture piece is big. And then our moist tropical. There's a lot of other subcategories where we find stocks, but these are our three biggest. And you can see, kind of like looking at this map, if the cool colors here are the big stocks you see in the northern latitudes, kind of in the tropical jungles, and then these coastal areas where we do have that high water content. If you look at the United States, you can kind of see that trend as well. We've got the Pacific Northwest, the South Coast, the Northeast, and this huge carbon pool in the north central United States, which developed under tallgrass prairie over the course of thousands of years. Again, we don't see it as we move west because we go from tallgrass prairie, where we have a mesic climate, to mixed grass prairie, which is drier, and then short grass prairie, which is dry before the mountains. So, all things considered, and there are a lot of caveats here with, again, kind of soil types, et cetera, but we see carbon accumulate in cool, wet conditions. Makes sense. Cool conditions kind of slow microbial processing. Wet conditions provide the ability for anaerobic preservation of carbon. Think about the peat bogs up in Ireland or Scotland as a perfect example. And then we see less carbon where things are hot and dry, and there's a lot of potential for oxidative loss back to the atmosphere. But soil carbon pools can vary extensively, even within relatively small geographic ranges. This is from Huang et al. Alfred Hardemink in the Soils Department was part of this study, where they used a model to map soil carbon stocks in Wisconsin. Here again, the cool colors are higher stocks and lower stocks. Taking into account this kind of important aspect of mineralogy, moisture, and temp, we see larger stocks in these southern prairie soils. Those are the mollisols, the soils that developed under tallgrass prairie. And we see these large stocks in our northern forests and wetlands. Again, we've got that cool, wet, and then we kind of have the high plant biomass input down here in the south. We see smaller stocks in the central sands, a lot of potential for oxidative loss, and in some of these deciduous forests, kind of in the middle band of the state. So, just want to say that those are how the stocks kind of look now in the 21st century, but that's not how they've looked over the last 12,000 years, which I know is a long time span, kind of since the last glacial maximum. This is a study that John Sanderman published in 2017 in the Proceedings of the National Academy of Sciences, where he basically used empirical data coupled with the Hyde model, kind of to look at the global distribution of crop and grazing land, which we see here. Cropland is our red land, grazing lands are our green lands, and the deficit over the last 12,000 years of carbon associated with those lands. And he came up with a map that looks something like this. It's hard for you to see, I'm sure, but this is soil organic carbon change in metric tons of carbon per hectare. Reds, the hot colors, are greater losses. Are increases. The cool colors are losses. And what he found is that over time, we see more carbon loss. It's going to sound counterintuitive initially. More carbon loss globally from grazing lands than from cropped lands. But the reason was that there's far more hectares of grassland that have been cultivated or managed over the last 12,000 years than cropland. When you look at a per hectare basis, the cropland was by far a much greater emitter or deficit of soil organic carbon. And ultimately, the number he came up with was that in the top 2 meters of soil globally, since the last ice age, we've burned off about 133 petagrams of carbon. So our soils now are not what we think they might have been at the end of the last ice age. This holds true locally too at a much shorter time scale in Wisconsin. We've been cultivating these soils for about 175 years. At the time, southern Wisconsin, which I'm going to use because we're here in southern Wisconsin as an example, most of the area was tall grass prairie or oak savanna. We came in first with oxen and then with steam tractors and broke the prairie or began to till the prairie. And estimates are hard to come by because the remnants that exist nowadays are remnants because they weren't cultivated for some reason. They're too rocky, they're too steep, they're abnormal. Do you know what I mean? They're not what the prairies were that were turned into farmland. But there are estimates out there for Wisconsin that show that we've lost somewhere between 15 and 20% of the soil organic carbon stocks in this area in the last 175 years. But that's only from the top 20 centimeters, so it's clearly an underestimation of the impact that cultivation has had on these soils. So the big question, and what people are excited about, is this idea of whether or not we can restore the debt that we've created through land management. This is actually some kind of generic spin-up data from the agri-IBIS model, which uses the century carbon model to predict carbon change. There's a spin-up phase in the model. This is kind of the way we think it might have happened, but for a couple thousand years, as prairies began to colonize Wisconsin, carbon accumulated. At some point, with a relatively stable climate, that carbon reached maybe an equilibrium. We don't exactly know, but probably some sort of pseudo steady state, this period here. And then in 1850, we came in with our moldboard plows and started flipping it all over and created kind of a mass exodus of carbon from the soil to the atmosphere. Question now is, if we, see the light of day and make major changes to our farming or management practices, can we develop future scenarios that put us back in a trajectory of accumulating carbon? The question is, what's going to help us build carbon, but then also, is it even possible to fill this gap? It might be, it might not be. So it's kind of a question and a real exciting potential at the same time. And that's why we see kind of all the excitement. I feel like when I started studying soil carbon, it was like kind of a nerdy thing. And now I just feel like it's like so overwhelming. It makes me sometimes want to change what I study because it's like a fire hose of stuff coming out about carbon constantly. But this is what we're after, this idea that if we can refill that gap, put that 133 petagrams back into the soil, it's coming from the atmosphere and it could potentially help mitigate some of the potential change in climate.
at the global scale, does erosion keep soil organic carbon in soil, or do you lose it?
That's a whole other huge kind of debate happening. I think the answer is yes. It depends on the outcome of the erosion, right? If the erosion creates deposition that then is inundated with water, you have carbon secured, or if it's somehow buried, carbon becomes secured. But if on the other hand, it exposes it to oxidative loss or more disruption. So it's really a yes, it depends. That's a classical answer, right? Academic answer. So how do we farm for carbon? I mean, at a really basic level, the idea is to increase carbon inputs and reduce carbon loss, kind of getting back to that NPP versus heterotrophic respiration. But there are some general principles that the scientific and kind of practitioner communities have kind of coalesced around. Things like minimizing soil disturbance. An example might be going no-till. Soil disturbance causes oxidative carbon loss. Keeping the soil covered, residue retention decreases or increases the albedo of the soil, so it actually cools the soil, keeps moisture in the soil. Maintain living roots, so growing more perennials, so we've got kind of photosynthetic carbon constantly being pumped down below ground, protecting ourselves against erosion. Maximizing diversity. I have a prairie here. I don't know if a lot of people are going to put their farm acres into prairie, but polycultures would fit into this. And then integrating livestock. This is a grazing system, but you could also imagine there's a lot of talk about people grazing cover crops. We work with some farmers that are grazing cover crops. So this idea of getting livestock back into a grain type of system. These are the big ones at a systems level that seem most promising. There's other things that people talk about, like fertilization. If we can increase above ground biomass and below ground biomass through fertilization, we have more carbon going into the system. Biochar is really fascinating. It's certainly part of the historic kind of prairie carbon story because those prairies burned. And then manure. There's a lot of studies that show that these things increase carbon. The kind of caveat here is, where did those sources come from? So fertilizer has a huge carbon footprint of its own. Biochar, if that biochar was generated on site, is clearly carbon from that site going into the soil, but if it's shipped from somewhere else, it's a carbon negative somewhere, and the carbon positive on your soil, and the same thing would be true for manure. If you take manure from one farm and spread it on another, it's going to build carbon on that farm you spread it on, but it's a export from the carbon you take it from. So these become more complicated when you start talking about it. There's a lot of literature on these practices, and a lot of that literature suggests that these are positives for soil carbon. There's also a lot of literature that says they're not positives, and there's also a lot of literature that says, we're not quite sure. And part of that is because of climate and soil, which we've already talked about, kind of the impact of those edaphic conditions on carbon. But I just want to throw this out there as kind of the conversation goes throughout the day, that part of it's also because of scientists, us using terminology incorrectly and kind of screwing up our methods occasionally. And so I just want to throw these ideas out there to kind of give you a sense of where I think some of the confusion is coming from if you dig into the literature. The first one is the impact of time when we start thinking about soil organic carbon sequestration. Let's pretend I create a study where I've started to do cover crops on a field. I'm going to call it a regenerative agricultural practice. And I run that study for 10 years from 2011 to 2021. And in 2021, I go in and assess soil carbon, and I assess it relative to a business as usual, like I wasn't putting cover crops or I wasn't going no-till. And that regenerative ag has more carbon than the business as usual. If I assume that the business as usual represents carbon at steady state, I might make the claim that the regenerative ag practice has sequestered carbon. Problem is that assuming steady state is a bad idea with soils because they tend to not be in steady state, particularly with a changing climate. So if we were to evaluate those two practices with a baseline carbon value, we might come up with an entirely different interpretation, which is that our business as usual has been losing carbon, and our regenerative ag practice has been kind of holding on to carbon. The use of space as a surrogate for time implementing practices and then looking at them after 30 or 40 years is really ubiquitous in the literature. And it's a great tool to say, you know, if you evaluated this in 2021, regardless of your interpretation, you would say that regenerative ag is doing better than business as usual. If you said regenerative ag was sequestering carbon, you would be wrong. And that's where some of the confusion with time, where longitudinal studies versus space for time studies come into play in the literature. The next one is mass, the importance of mass when we understand carbon. If we sample in 2011 a bunch of soils at 0 to 10 centimeters, and then we implement this regenerative ag practice, and that regenerative ag practice results in a decrease in bulk density, soil organic carbon increases, the soil gets fluffier. We come back in 2011 and take our soil cores again, we're now sampling a fluffier soil. So our core that went down to 10 centimeters in the first year is not going to go down to that same 10 centimeters in the 10th year. It might go down to 8 centimeters. And so we end up evaluating carbon pools from 2 different masses of soil. It's very likely that the carbon concentration is going to be different, but it's not actually correct that that carbon difference is because of the treatment. It's a combination of a treatment effect and a change in bulk density. And so there are a lot of methods out there to correct for those changes in bulk density, equivalent soil mass corrections, there's R code, there's all sorts of resources out there to do it, but there is a huge body of literature, extant literature out there that we rely on to understand carbon sequestration that does not make these corrections. And finally, depth is something that's worth considering. The IPCC recommends measuring carbon to a depth of 30 centimeters. Most studies out there study carbon anywhere from the top 2 centimeters to that 30 centimeter horizon. And the problem with that, I mean, the reason for doing that is that that's carbon that's easy to measure, it's easy to sample. It's the area of the soil that's most affected by our management practices. But the problem with it is that it's not the entire story happening in any given soil. So this is just an example from Arlington at Wixt. If this is 0 to 15 centimeters and 15 to 30 centimeters, up at Arlington, that's about 60% of the carbon in the soil. It's a partial picture of what's happening. But 40% of the carbon is happening below that. And our studies have shown that we're losing carbon in these lower horizons, in some cases offsetting any kind of gains we're making in the surface. So collectively, I think these kind of methodological issues have complicated some of what we're seeing. And in fact, a recent study by Don et al. This year in Global Change Biology found that of 100 recent studies on carbon sequestration in soils, only 4% of them, or I'm sorry, only 4%, so 4 out of 100 used those terms correctly. The others were misattributing, space for time or lack of equivalent soil mass corrections to things like carbon sequestration, which they are not. So I'm probably talking too much. So what does this look like in practice? This is new data from the Wisconsin Integrated Cropping Systems trial, a 34-year trial up at Arlington. We've gotten to the point where we're assessing these soils kind of on a decadal time step. And we took the opportunity this time to look at the impact of these kind of methodological considerations on what our take-home message would be, primarily because of all of the carbon accounting and carbon trading schemes that are coming out, and the fact that they all have different protocols and methods requirements. So let me just show you, because all of the figures I'm going to show you are going to be the same. We've got megagrams per hectare per year. This is actually a delta, a change over that 30-year period. We have the systems at Wixt: continuous corn, no-till corn, soybean, organic corn, soy, wheat, dairy forage with corn and alfalfa and manure, organic forage with corn and alfalfa manure, rotationally grazed pasture, and a prairie. This 0 line is indicating no change in the past 30 years. The whiskers are 95% confidence intervals. So if the whiskers cross the 0 line, they're not statistically distinguishable. And then your little letters indicate significant difference between treatments and change. So If we were to go in there and use space for time, well, we'd first have to pick what the historical management would be. And in this case, it would be the conventional forage. That was what WIXT was before we created the trial. I didn't create the trial. Josh Poser created the trial. And if we did that using this forage as kind of our baseline, we would interpret these results as pasture sequestering carbon, forages and prairies stabilizing carbon, and all of our grain systems losing carbon. If we came in here and looked just at our soil sample depth and didn't correct for changes in bulk density, we would say that our organic grain and our pasture have both sequestered carbon and that all other systems are stable or holding on to carbon. If we look just at that top horizon, the top 15 centimeters, 6 inches, which is where most people sample soils, we would say that our grasslands have sequestered carbon, our organic grain and our forages have maintained carbon, and our continuous corn and no-till corn soybean have lost carbon. But if we evaluate things comprehensively, taking the whole horizon into account, correcting for soil mass changes, bulk density changes, and using a longitudinal approach, we see that our grasslands, prairie and pasture are holding on to carbon. There's a lot of variability there, but they're kind of maintaining carbon. And all of our other systems that would be considered field crops, forages with manure, organic grain, no-till, and continuous corn have all lost carbon. And it's this comprehensive assessment that is the closest to what's actually happening on that soil. Yeah.
Can you explain, Greg, anything with, I see the distribution of your sample is much larger in the comprehensive compared to the other ones. Is there any inclination of why that is?
Well, part of it is because in this case we have, compared to here, we have 4 soil depths instead of 2 soil depths, which introduces a bunch of variability. But then I guess I'm not entirely sure. I'd have to, this was Claire, our grad student, my master's student, and I did this. I think it's in part because of the fact that we've got 4 horizons going into this one, but also the fact that different systems have different number of plots in each block, which is a little bit of a weird statistical design part of WIXT, but that's part of the story. Not a complete answer, but I can get you the complete answer. Yeah.
So the prairie treatment in this case had no animals on it, other than the micro.
In this case, we're just talking about our control prairie. We're about to put out a publication on just an assessment of our prairie, because our prairies, we do have prairies that are harvested for bioenergy, prairies that are treated as a prairie restoration where we just burn them every couple of years, and then prairies that we actually do graze. With the same cattle, during the summer slump of our cool season pastures, we move the cattle in through the prairies, and we're about to work those numbers up.
Right.
Yes.
Definition semantics means something. So using the term sequestration implies the WRI standards of permanence.
Mm-hmm.
You and I talked about there's a lot of varying degrees of how soil carbon is stored in the soil and
Sure.
its stability. Can you comment on stability in terms of?
I am gonna hit on stability.
Okay. All right.
Yeah, that's a great question. I think I'm probably gonna be over time, so I'm trying to get through some stuff. So in terms of what are realistic sequestration rates, in addition to kind of the excitement about sequestering carbon, there's also a lot of kind of big claims being made. And so I just wanted to kind of ask the question with a couple studies from our part of the world, the north central US, kind of maybe what we could expect. So this is the WIXT data, but it's from the 20-year time step. The numbers actually fall in very close to the 30-year time step. Here I've grouped it more by kind of management. So prairie, CRP, which is a lower diversity prairie, pasture, forage, and grains. Same interpretation. In this place, these are 90% confidence limits. And these are younger systems at the time, so 10 years versus 20 years. And what we saw is that in the WIXT systems after the first 20 years, carbon sequestration kind of went from losing 1.5 metric tons of carbon per hectare per year to about 0, like being stable. In a similar study, which has not yet been published, unfortunately, from colleagues at the Kellogg Biological Station in Hickory Corners, Michigan, where they looked at no-till, reduced input, biologically based, this is kind of akin to organic, and alfalfa over a 20-year period. You know, a similar scale here, 0 is no change. The bars, in this case, equal standard deviations, and the letters equal significant difference. They saw in all of their systems an accumulation of carbon over that 25-year period. So what's going on? A lot of things, but one of the main things, I think, is the fact that the data from Wisconsin comes from a prairie soil, a mollisol, and the data from Kellogg Biological Station comes from an alpha sol, a forest soil. But it's not just that. The texture at KBS is much coarser. There's a lot more sand. There's a lot less potential to actually stabilize the carbon that's being put into the system, but there's also a lot lower original carbon content. So here we're starting off really high and trying to build it. Here, they're starting off much lower and actually have been able to build that carbon in those systems. So, you know, they're ending up in the 0.2 to 0.7 metric tons of carbon per hectare per year. So I feel like it's realistic to think about plus or minus a half a ton of carbon per hectare per year. There are going to be areas where that's positive, and maybe even beyond that, a ton of carbon per hectare per year. And there are going to be areas, whether we like it or not, where it's less than that. We're losing carbon every year. So we're hovering around a mean, and it's not all that surprising, given the fact, going very back to the beginning of the presentation, that there's this constant flux, right? CO2 is being pulled in by plants, microbes are working on the carbon and spitting it out, and that's happening constantly. So in any given year, you're hovering around 0, and it's the long-term accumulation of years where you're up above 0 that slowly builds that. Just to point out again, you know, this is prairie, this is forest, coarse textured, fine textured. If you look at a map of the United States and our soils, the greens and blues are our tall grass prairie soils. Those are mollisols. In this case, they're udols, the green soils, and aquols, the blue soils. And then here we have eustols. Those are our mixed grass prairie mollisols. All of these are prairie-derived soils, so they're high in carbon. By definition, they have 35% or more of their base, of their cation exchange sites saturated with base cations. They're super fertile soils. And because they're so super fertile, we grow a lot of crops here. A lot of corn, a lot of soybeans, a lot of tillage, a lot of, you know, management that doesn't incorporate those management practices we talked about. And so you overlay the Corn Belt on top of this huge carbon reservoir, and it's a tricky situation to think about managing. So again, there's going to be places where it works, places where it doesn't, but I just want to throw that out. Like, it's not, the results from WIXT aren't so anomalous when you think about the fields that we're managing in the north central US. In terms of sequestration, kind of like how long will the sink last and how long will the carbon we store be permanent? Getting to your question. This is a little bit of a sophomoric figure, I apologize, but this is from a Nature article just looking at not saturation, but the slowing of carbon accumulation. And most publications that talk about using carbon, soil carbon as a natural climate solution do suggest that, you know, a change in practice, let's say from continuous corn to prairie, that would be a huge one. And that might not be a good example, but let's say from continuous corn to no-till continuous corn with a cover crop. That tends to result in about 20 to 50 years of carbon accumulation before we start stabilizing. I'm not going to use the word saturation because I don't know that I agree with that. And we're still far below that deficit of the prairie at this point, but the easily built carbon has happened, right? It took the prairies 3,000 years. In 20 to 50 years, you build up your particulate organic matter stock, and then it begins kind of a long slog of slow development of that soil. So these sinks are pretty active for 20 to 50 years, at which point they tend to kind of slow down. And here's the tricky part. It's really easy to burn that carbon off. Again, in 175 years, we lost 20+% of the carbon in our soils from cultivation. Carbon is stabilized in soils either because it's physically disconnected, again, occluded in aggregates, it's sorbed to clay mineral surfaces. And these stabilization mechanisms are great because they hold on to carbon, but if I were this farmer that decided to go no-till and plant cover crops, and I retired and handed my farm to my son or my daughter, and they decided that they really liked moldboard plows. Within a decade, almost all of the gains that had been made would be gone. So the idea, I guess one of the big pushes that I've always had is we need to protect the carbon stocks we have as kind of a first line of defense, and then see where we can build carbon. And once we build, find ways to protect the carbon stocks that we build. Does that help a little bit? Maybe not. We can talk later.
Because how much longer?
I'm going to be done in a second. I'm so
Okay.
sorry. Thank you. So takeaways. Soil organic carbon is huge for soil health and function. Key indicator. It has the potential to slow climate change and provide all sorts of other ecosystem benefits. We know how to manage soils to move them in the right direction. Whether or not that means sequestration is uncertain, and that comes down to kind of, I think, a lot of expectations confounded by misattribution of data. Data that doesn't really show sequestration, that we're interpreting as sequestration, has kind of confused the story a little bit. But we know that if we can minimize disturbance, maximize the amount of plant biomass going in, we can move carbon stocks in the right direction. And then again, there's plenty of co-benefits that make building carbon a good idea above and beyond any kind of climate impact. Okay, that's all.
Thank you.
Thank you, sorry.